Okay, that's fine. Good afternoon, colleagues. Good afternoon, colleagues. Thank you very much for joining us for the APN meeting for the month of May. I think we have enough people and we can start. Other people will just join us along the way. It's recorded and posted somewhere, so I'm sure I'll be told where it's posted. So for those who are late, you can see the first part of the meeting later. So today, we're very happy to be getting a presentation from Professor Phumla Sinxadi. She's a holder of an MBCHB, a diploma in anesthetics, an MEd, and a PhD. And she's a clinical pharmacologist and the head of division for clinical pharmacology in the Department of Medicine at UCT, in the Faculty of Medicine. She is the deputy director of a newly established South African Medical Research Council, UCT, platform for pharmacogenomic research and translational unit. And she has more than a decade of experience in convening and teaching undergraduate pharmacology and courses for medical students and postgraduate teaching and supervision. She also has a National Research Fellowship C2 rating as a scientist, and the research focuses on investigating concentration response relationships between antiretroviral drugs, toxicity, and how genetic differences influence drug exposure and clinical outcomes. So today she's going to be presenting a topic on HIV pharmacogenetics. Professor, I hand over to you. Thank you very much for making time to make this presentation to the network.
Thank you so much, Tinashe, for the kind introduction. And thank you, Nosipo, for inviting me to your seminar. Can you guys hear me okay?
Loud and clear.
Great.
You can go ahead.
I've slightly tweaked my talk and I wanted to make it a little bit more focused. And so I've entitled it Pharmacogenetics of Modern Antiretrovirals in Southern Africans, to actually illustrate that my work is mainly focused in our population. Okay, so as we know that sub-Saharan Africa carries the largest burden of HIV epidemic, with East and Southern Africa having more than 20 million people living with HIV. In South Africa, we have the largest antiretroviral program in the world, and programmatically, we actually followed the WHO guidelines and switched from the efavirenz-based antiretroviral therapy to dolutegravir-based first-line regimen around 2019. Dolutegravir is a second-generation integrase strand inhibitor, which is mostly metabolized by UGT1A1. Some of the advantages of using dolutegravir include having fewer drug-drug interactions, having a higher genetic barrier to resistance, and having a better safety profile, particularly when you're referring to neuropsychiatric side effects, which efavirenz was notorious for. However, one of the emerging concerns with the use of dolutegravir is the marked weight gain, and this has been a focus of some of the work that I'm going to be presenting today. With the HIV regimen, currently the nucleoside or nucleotide reverse transcriptase backbone includes tenofovir prodrug, either including tenofovir disoproxil fumarate or TDF, or tenofovir aliphenamide or TAF. I'll be referring to TDF or TAF in the future slides. And these are extensively prescribed. In South Africa, in particular, we mainly prescribe TDF, and in private, there are people who have access to TAF. One of the concerns with using TDF is that it's associated with more renal and bone toxicity than TAF. And between the two prodrugs, the main difference is that the TDF actually, in terms of exposure or pharmacokinetics, it yields 10 times more plasma tenofovir than TAF, which is also thought to be associated with its toxicity, while TAF, on the other hand, actually has more of the intracellular metabolite, which is active metabolite, which is TDF, tenofovir diphosphate. So I'm going to mainly talk about the weight gain as the outcome for this talk. And so I will be referring to that more, but I'm happy to take questions that are not related to the talk at the end of this talk if we have a lot of time. So with initiation of antiretrovirals, we know that this often leads to weight gain. While some of this weight gain is an appropriate return to health effect, excessive increases may lead to obesity. And additional factors that are associated with weight gain include demographics, which can include a black race or female sex, and HIV-related factors. These will usually include high pretreatment baseline viral load or the low CD4 count at baseline. The third, or the important one for this talk, is the choice of an antiretroviral regimen. We have integrase inhibitors and tenofovir aliphenamide being the main culprits when it comes to weight gain. In the graphs that are displayed on your screen, we have both the weight change or the absolute weight graphically represented. And you can see if you're looking at different drug classes, if you're looking at panel A, we can note that integrase inhibitors actually increase more weight gain. I don't know if you can see my arrow. So the instis cause more weight gain than the PIs or the NNRTIs. And if you're looking with between the instis, bictegravir and dolutegravir cause more weight gain than elvitegravir. And then if you're looking at NNRTIs as a class, then you can appreciate that rilpivirine causes more weight gain than the efavirenz arm. And then if you're looking at NRTIs, TAF is the one that actually causes more weight gain. And this is seen whether you're looking at weight gain as an weight change or looking at the absolute weight increase. The mechanisms, however, of how these antiretrovirals cause weight gain are incompletely understood. So part of the work that I do, like Tinashe said in the introduction, is trying to look at concentration outcome relationships with antiretrovirals and trying to look at how genetics actually affect the individual variability with exposure or the manifestation of the adverse effects. So we teamed up with Professor Francois Fenter from Ezinja, based at Wits. He's the PI for the advanced randomized clinical trial. And we were trying to answer some of the critical questions around dolutegravir, TAF, and weight gain. And so in brief, this work has been published already, and they've looked at 3 arms in over 1,000 participants living in Johannesburg. And the 3 arms were comparing TAF, FTC, and dolutegravir, TDF, FTC, and dolutegravir. And these 2 arms on dolutegravir were actually compared to standard of care. And participants were followed up from baseline to 192 weeks. And if you look at the graph on the right side of the screen, you can see that in all participants, when someone starts on antiretrovirals, they actually do gain some weight. And I've spoken about this before as maybe a return to health phenomenon. But what's important to note is that if you were in the TAF FTC dolutegravir arm, you gained more weight than if you were in the TDF FTC dolutegravir arm, and even it was even the difference was bigger with the TAF arm, and it's actually even more appreciated in women, like we said with the initial slide that was talking about the weight gain and ART. So this the graph is actually showing up to week 96, but the publications reporting up to week 92 show that this weight gain is actually not plateauing. It's sustained over the 192 weeks. So it is an ongoing concern being on TAF and DTG when it comes to weight gain. So we aim to identify some genetic polymorphisms that are associated with individual differences in drug exposure, efficacy, and toxicity of dolutegravir and tenofovir. I'm going to focus mainly on objective 3 and 4, but objective 1 was to look at SNPs that affect tenofovir clearance, and objective 2 to look at SNPs that predict tenofovir associated renal toxicity. Both those objectives have been looked for and answered through a master's degree, which was done by Somila Mateza and Zintle Cindi. And I will, if somebody is interested, I will actually, I can forward the publications, but it's already published. The same with objective 3 and 4, looking at SNPs that predict increased dolutegravir plasma exposure, as well as looking at dolutegravir associated weight gain. But for this talk, I'm only just going to focus on objective 3 and 4. With this study, the methods, depending on the objective, were similar. So we had looked at pharmacokinetic analysis. We conducted pharmacokinetics modeling with the UCT pharmacometrics group, and then the genotyping and imputation, and then the analysis. So with the PK analysis, we actually looked at 2 different ways. There was subset of patients who underwent intensive PK sampling, and the rest of the cohorts had sparse sampling. So we used pharmacokinetic PK modeling to actually get exposure for everyone who was included in the cohorts. The PK analysis were done using liquid chromatography with tandem mass spectrometry, or LCMS-MS, and all that was done in our laboratory, our PK research lab. And Professor Lube Wisner is the director for that lab. So I'm just also advertising, if you want PK work done, we have a lab to do it. And then with PK modeling, they use non-MEM, or nonlinear mixed effect modeling. And our outcome of exposure, the outcome of interest, sorry, was the unexplained variability in the dolutegravir exposure. And we looked at the AUC and variability in the AUC, the actual AUC, and then we also looked the between subject variability. And we wanted to look at that because we wanted to look at what was explaining the difference outside of the other demographics. So age, weight was already accounted for during the modeling. So the only thing that we regressed with the outcome was just the sorry, there's something that's appearing on my screen. It was just the regression between the unexplained variability. So not adjusting for the age, sex, weight again, which were already included in the modeling. And then with the genotyping, we collaborate quite extensively over a long period of time with Professor David Haas at Vanderbilt. So our samples were shipped to Vantage, and the Illumina Mega X chip was used for the genotyping. The data was imputed with TopMed, and we use Plink 1.9 for the analysis. And then when we looked at, or when we were doing the genetic analysis, we had similar approach depending on the outcome of interest. But basically, we had three approaches. Firstly, we looked at associations with the SNPs that have been previously associated with the dolutegravir disposition. Secondly, we looked. So firstly, actually, it was functional SNPs that are known. Secondly, we looked at the genes and that are associated with the disposition of dolutegravir. And then we looked at PharmGKB and the GWAS catalog to look at SNPs that may be of interest if they were previously associated with any trait. And finally, we explored associations genome-wide. So the approach was similar, and I'm only going to present the results from the objectives 3 and 4. If there's anything that I need to explain afterwards, I'll be happy to take questions later. So just to give you an idea about the population, this was just the baseline characteristics for the weight gain associations cohort. And what's important to note is that we had more than 700 patients on dolutegravir arms. However, only about 48% of the cohort gave consent to genetic testing. So our final analysis cohort is much smaller because more than half of the population didn't consent to genetic testing. And what's important because this was a randomized clinical trial, was that there were no differences in terms of baseline characteristics, whether you're looking at age, sex, baseline BMI, weight gain at week 48, and the nadir CD4 count and viral load when it comes to those who consented and those that didn't consent. So I don't think there was some systematic bias based on the people that did or did not consent for genetic testing. So with looking at genetic associations with weight gain, we looked at the whole population, so everyone in one and together, and also we split whether they were on looking at participants that were on the TDF arm and participants that were on the TAF arm. And we actually found when we looked at genetic associations focusing on the SNPs relevant to dolutegravir or tenofovir disposition, the SNP with the lowest lowest p-value was a SNP in ABCG2, which had a p-value of 0.03, which after adjusting for multiple comparisons didn't meet the threshold for significance. We also looked at the genes of interest for tenofovir. For that analysis, when we looked at those genes, as the lowest p-value was in ABCC10, but it also did not meet the threshold for significance. When we looked at genome-wide analysis for weight gain at 48 weeks, we found only one polymorphism reached genome-wide significance, which was in TMEM163. And this is a transmembrane protein 163. And to our knowledge, this has never been reported before. And the only thing that we know about this polymorphism is that it was previously associated with insulin resistance and weight circumference. But there was a different SNP that in that protein that was significant, but it wasn't the same SNP that we found. So we think maybe there's something there, but this would need to be replicated in another study. And to our knowledge, it was the first time that we had looked for an association between dolutegravir and tenofovir associated with gain, and in SNPs that are relevant to their disposition. Next, I want to talk about the associations between genetic associations with exposure to dolutegravir. This was, again, in collaboration with David Haas, and this was work that was done by Zinhle, who's now a postdoc at UPenn, University of Pennsylvania, under Professor Ricci. Zinhle worked with Ida, who was also a PhD student in the division. She's now at in Uganda, back home in Uganda, as a postdoc. And they collaborated with this type of work. So Ida did the POP PK, and Zinhle did the genetic analysis. Again, our initial focus was on known polymorphism, and then we looked at the UGT1A1 gene because, like I said before, the metabolism of dolutegravir is mainly through UGT1A1, and there's a minor pathway with CYP3A4. So our initial focus was on the functional polymorphisms, and then we looked at UGT1 gene, and then we explored the associations genome-wide. We actually did find, if you look at the table, it's actually looking at the SNPs of interest, and our the top SNP was the SNP that we were actually particularly interested in, and it was significant with a p-value of 1.8 times 10 to the power minus 4. And we only included 284 participants, which is a further smaller cohort because not everybody who had consented to genetic testing had PK results. And then we found a statistically significant association with UGT1 RS887829 and the variability in the dolutegravir AUC. And then we used the genotype data in the final model for the dolutegravir exposure. And if you look at the, I'm trying to find the laser pointer. I'm not sure if you can see my arrow. But anyway, if you look at the concentration time graph, which was simulated, it shows that if you carried a CC genotype and you had, if you carried a CC genotype, there was more effective clearance compared to homozygous CT genotype or, heterozygous CT and homozygous TT genotype. So the clearance was decreasing for CT and TT. And you can see here participants with TT genotype had higher exposure. But what's important for, I suppose, clinicians is that if you look at the concentration where there is virologic prevention of virological replication, even the participants who carried the CEC genotype had much higher concentration than what is needed. So it's reassuring that even if you have, there's no worrying about antiviral efficacy, even if you can metabolize dolutegravir effectively, but we may need to worry about toxicity in patients who are carrying the TT genotype. We also looked at like I mentioned, we also looked at dolutegravir exposure genome-wide. And these Manhattan plots are actually showing in panel A the dolutegravir exposure, and in panel B, we used bilirubin as a biomarker for UGT1A1 activity because it's conjugated by the same enzyme. And in panel A, we show that I'm trying to find the pointer. Sorry again. So in panel A, you can see that the lowest p-value actually had a p-value of just under 10 to the power minus 8. And it was a SNP in CAM-KMT, which is related to calmodulin and has not been previously associated with dolutegravir pharmacokinetics, and it's unlikely to be a true association with dolutegravir pharmacokinetics. And then in panel B, we looked at bilirubin or logged bilirubin concentrations, and the lowest p-value was RS6742078, which was within the UGT1A locus, and the p-value was 7 times 10 to the power minus 13. And importantly, this polymorphism was in strong disequilibrium with our SNP of interest, which is RS887829. At the end of the day, you have to wonder, does it actually matter if there are genetic associations with concentrations clinically? So one of the questions that we also asked within this study, I'm trying to move, was looking at whether loss of function polymorphisms predict the failure of weight gain in patients who are on efavirenz. And for this analysis, this was work done by Rulan, who just finished his PhD. And we actually investigated whether people who were slow metabolizers of efavirenz were gaining more weight, because there was a study that showed that patients switching from efavirenz to dolutegravir, and which was something that was being done programmatically, those patients who were slow metabolizers gained more weight. And when we looked in our cohort, we have the red, green, and turquoise lines representing the extensive intermediate and slow metabolizers. And then the blue is patients who were on dolutegravir. And if you look in both men and women, the extensive metabolizers showed a weight increase, the same as the patients who were in the dolutegravir arms compared to the intermediate or the slow metabolizers. The slow metabolizers actually showed loss of weight because they were continuing to be in the efavirenz arm. And these findings actually suggest that the weight gain differences between efavirenz and dolutegravir are driven by impaired weight gain due to efavirenz toxicity rather than by off-target effects of dolutegravir causing weight gain. So we still can't explain why patients on dolutegravir are gaining weight because we haven't established that there is a causal relationship between using dolutegravir and gaining weight. We further looked at whether this is driven by concentrations, and in this graph, we're looking at the correlation between efavirenz mid-dose concentrations and weight gain. And in the first graph, we are actually showing that there is a negative correlation. If you have higher concentrations of efavirenz, you tended to lose more weight. And we saw the same thing when we looked at the correlation with dolutegravir, which was unexpected because we didn't think there was a pharmacological explanation for dolutegravir causing weight loss, because most people on dolutegravir actually gain weight. But when we adjusted for age, sex, BMI, the association with dolutegravir actually was not maintained. But with the efavirenz association, even when you actually adjusted for age, sex, CD4 count and viral load, pardon me. So it was age, sex, CD4 count and viral load. You could still appreciate that there was an association with weight loss and having higher concentrations. So we can at least say that when you are on dolutegravir, gaining weight still needs to be explored about why patients gain weight on dolutegravir, because there has been a positive association that's been explained. And it's been seen again with patients who are placed on TAF versus TDF, and that's also been explained through the toxicity of TDF or the toxicity of tenofovir on weight and in impairing weight gain. And then the last thing we have to worry about was whether if dolutegravir, which is the newer antiretroviral, is causing weight gain, should we be adjusting these doses in patients who are obese? And what we did was look at a subset of patients who underwent intensive PK sampling, and we were looking at the exposure of dolutegravir, and we compared patients who were obese versus patients who were not obese, or patients without obesity. And we can appreciate that the exposure in patients who were obese was marginally lower, but this is unlikely to be clinically significant because, again, even in patients who are obese, we get concentrations that are much higher than the minimum that's required for preventing viral replication. So we don't need to be adjusting doses in patients who are on dolutegravir and are obese. Right. So my last slide is basically a summary of what we've done with the dolutegravir exposure and genetic analysis. We know that there is greater weight gain with dolutegravir and TAF, and that is also been shown in the advanced cohort that the patients who are female have an increased risk of excessive weight gain. We couldn't adjust for race because most of the patients that were our participants, that were enrolled to the advanced cohort were mostly Black. And then with the weight gain differences, the weight gain can be explained by the efavirenz toxicity. We've shown that if you are a slow metabolizer of CYP2B6 and had higher concentrations, there's failure to gain weight if you start on efavirenz-based ART. But for patients who are switching from efavirenz to integrase inhibitor, if you're a slow metabolizer, you then gain more weight. When it comes to exposure to dolutegravir, UGT1, we actually were able to confirm that UGT1A1 polymorphism, the RS887829, was associated with increased exposure. And when we used the bilirubin as a biomarker, that also confirmed that was a true association. When it comes to weight gain, we couldn't find an association between UGT1A1 and weight gain, and we found a novel genome-wide significant polymorphism that still needs to be replicated. I just want to acknowledge all the collaborators that I've worked with. I won't list them. And this funding was made possible by the NIH before the troubles that we're experiencing now. Some by the Wellcome Trust or the SAMRC, the NRF, and ViiV, some of the pharmaceutical companies, so ViiV and Gilead, and Unitate and USAID. And then we've had local collaborators within the division, as well as collaborators with Franchofenta, which has been a fruitful collaboration. We've produced several publications and several postgraduate degrees doing this work. Yeah, I think that's it. Thank you very much.
Thank you very much, Prof. Thank you very much for that very detailed presentation. And I like the last slide that you had on the presentation, which is the same theme as one of your papers, which was about dolutegravir, which was association is not the same as causation.
Oh,
I think that was published.
You that we wrote, yes.
Yes, yeah. Yeah, I think you were really trying to make sure everyone understood that there is maybe no causation here. Yeah. So, and if anyone has a question for Prof, you can put it in the chat box, or you can raise your hand. We can ask you to unmute, and you can ask a question away. Try and keep the questions to a few, so that we can allow as many people as possible. And I think Prof Colette is. Yeah, that'll be fine if you don't want to refer back to them in some of the answers. So Prof Colette is asking, why do you say that UGT1A1 SNP was unlikely to be associated with dolutegravir toxicity?
So when we looked at, we actually did a genome-wide association with weight gain, and it didn't make there was no association between weight gain and UGT1A1. Is that the question you're asking, Colette?
Not really. I'm asking where you have got your two Manhattan plots, and in one of
All right.
your code, you have got two signals, and the other one is one, and you quickly discounted the other one. That is, it's not relevant. And
so I know which one you're talking about. So the panel
this one,
A,
yeah.
yes. You guys are still seeing the big slide, right?
Yeah.
Okay.
Yes, sir.
So with the top SNP, there is not in UGT1A1. It's still in chromosome 2, but it was a CAM-KMT polymorphism, which is which is involved in calmodulin pathway. So nothing to do with dolutegravir exposure, not that we know of. And we actually looked if there was any meaning to it, and we found that the previously polymorphisms in that gene had been associated with anxiety. So couldn't explain the dolutegravir exposure.
So, Prof, don't you think that the limitation is our understanding of what we think should be affecting DTG, don't you think this opens up an opportunity for us to go deeper and say, maybe when you look at linkage of genes and all those pathways, it could be associated through the gene networks?
So, I mean, we have to look at biological plausibility, and that's what we found lacking between how the dolutegravir is metabolized and what is the function of this gene.
Okay. Thank you.
Okay. There was another hand.
So, yeah, I'll read one of the questions in the chat box for now, and we'll take the hand next.
Okay.
So there's a question from Lisa. Do you have a hypothesis why more than half the study participants did not consent to genetic testing? Do you have any idea how to inspire a different perspective on genetic testing? I think that's looking at the uptake by patients there.
Yeah. So I think we actually explored this quite a bit, and what transpired from discussions with the investigators at the site was that they actually first asked participants if they were consenting to having their blood taken for storage for later use. And a lot of them said no. And if you said no to having your blood drawn and stored, then they didn't ask for consent for genetic testing. Patients were happy to have their bloods taken to look for safety, but they weren't happy for having their blood taken to be stored and to be used at a later stage. So that was one of the, I think it was how the patients were approached in a sense when it came to genetic testing. That's the reason why patients said no. They just wanted to check whether their the kidneys were functional okay, but it wasn't a fear of whether doing genetic testing they're going to be cloned or anything like that. It was mostly that if they were asked whether they would agree to storage, if they said no, then they were not further asked whether they wanted, they were not asked specifically whether they wanted to say no or yes to genetic testing.
So it was initially a question on biobanking, and then that would deprive you of the genetic test as a result of that.
Yes.
Okay. So I have a question on the issue of weight gain on ARVs, not just the dolutegravir, on any of the previous studies you've done. The weight gain, does it translate to metabolic changes that result in adverse cardiovascular disease risk, lipid derangements, and other diseases that come with that? Does it actually translate to that? Because I noticed at the initiation, all three groups that you had in the study, I think there was the median BMI was actually normal. I think it was 23 for almost all participants,
so
23 point something.
Yes, for when participants started, they had a healthy BMI. Yes, that's baseline at baseline. So the advanced group, the main papers actually do speak on this, and they did look at whether patients changed categories from being healthy weight to being overweight and then obese, and it did show that there was a change in percentage of people moving between the categories. And the other concern was whether there are issues with increasing blood pressure and other cardiovascular risks with dolutegravir use. There was a signal that was reported in one of the CRO conferences, but I don't think we have replicated that in this cohort. When they looked at the blood pressure, people didn't have further metabolic complications or increase in insulin resistance, for example. But the weight gain issue was something that was sustained.
Well, was lipid profiles part of the endpoint? Were lipid profiles part of the endpoint? Cholesterol levels.
I'm trying to remember. I think they did actually. It is part of the safety monitoring, and I can't remember if there was a particular concern for dolutegravir, but we've looked at I didn't present it today, but we've looked at associations in patients who are on efavirenz with the efavirenz genotype, and we've shown that you usually have an increase in total cholesterol and the HDL cholesterol, but we didn't actually find I don't know if she's actually joined, one of my students, Jessica, recently published a paper on the efavirenz and the outcomes, including the lipid profile, ALT, and trying to think. Yeah, it's the lipid profile and the liver the ALT, which was one of the outcomes that we looked at, and glucose. And we didn't find an association with glucose or triglycerides. We found with total cholesterol, but it was only limited to the efavirenz arm. I can't tell you offhand if they actually found any associations in the dolutegravir group.
Thank you, Prof. Anyone else with a question? I'm not seeing anything in the chat. You can raise your hand and we can ask you to just unmute and ask directly.
Yeah, I have a question. Thank you very much. It's a very, very good presentation. Something on your conclusion caught my eye. You said black race increased risk, or so I wasn't sure. You are referring.
This is not this wasn't referring to this cohort in particular, but if in the introduction I did mention that there have been cohorts that have looked at weight gain when you initiating antiretrovirals, and there has been an association with black race and increased weight gain. Sorry,
Yeah,
I didn't let you ask your question. Apologies.
No, that's helpful. But the whole idea I was trying to understand is it's not race that is causing. It's just some other unmeasured or other confounders. So, so the implication what you are saying is black race is genetically different from others, and it's causing the biology is different. Basically, you are saying, but it could be due to others than just the self-reported race. So I just want to make it like, whether you are saying race is a biology or not.
It is there is association, but I'm not saying it's causal.
Yeah, but that association could be unmeasured factor or, others. Yeah, thank you.
Thank you for that question. I'm not seeing any other questions. Anyone else with a question, please go ahead and mute.
I don't have an access to I don't see the chat. Maybe I need to stop sharing. Then
no, I'm paying attention to it. There aren't any new ones.
So the other question I have, I can add one more, is you have a very small sample size. And yet I don't see you have a plan to replicate, but saying those are really important SNPs or causal. Have you replicated your finding? Because you already mentioned association is not causation. So why do you believe these variants are important without replication?
So which ones in particular?
I was talking in a very general sense. Have you replicated your finding?
It is. I mean, we haven't replicated in because this is data from the advanced clinical trial. So We haven't replicated any of the findings that we in a different cohort. But I think we fairly confident that the association between UGT1A1, 887, 829, and dolutegravir polymorph and dolutegravir exposure is true, because there has been it's not a novel association. We're not the one who are discovering it. We are replicating what has been previously described for that polymorphism. What we don't know is the one that we found with on the GWAS between TMEM 163 polymorphism and weight gain. That was a novel finding. It hasn't been replicated by anybody else. And we don't have a different cohort where we could replicate those findings. And we agree that these numbers are small, but I think I'll be happy if somebody else actually does replicate it. It could be beginner's luck.
So, Prof, this is an important thing. We're seeing quite a number of papers coming from your lab around the advanced clinical trial. What can you say to us about the advantage of attaching a pharmacogenomic study to a clinical trial, especially with some of the more cleaner endpoints being measured more accurately, rather than some of us using real-world retrospective data, which at times can mask some of these findings? I don't know what's your comment on attaching a pharmacogenomic study to an ongoing clinical trial like that.
I think it is a strength. I found it's a cheaper way of doing pharmacogenomics, because the main study is already paying for recruitment of patients, and usually for doing a pharmacogenomic study is a matter of getting ethics approval and them storing an extra tube of blood for genetics. But also when you have well-characterized outcomes, it's better than doing a retrospective study, because then you can accurately document the outcome of interest prospectively. I don't know if that answers your question. I think I feel it's an efficient way of doing it, rather than starting from scratch or looking at outcomes where you haven't controlled for how the outcomes are going to be measured beforehand, because there could be variation in the definition of or characterizing your outcomes of interest. And if you haven't collected in the same way, that introduces another source of variability in your analysis.
Thank you very much. There's a question from Prof. Colette. Are we saying DTG is associated with weight gain, but that we are not sure about the mechanism or whether it's genetic, or the genetics of that link?
Yes,
question from Prof. Colette.
We haven't determined how dolutegravir increases. We haven't answered the question about what is the mechanism behind dolutegravir associated weight gain.
But we confirm that it causes weight gain. It is associated with weight gain.
It's associated. Not
yes. Okay. So the association is there. Okay.
The association is there. We're not denying the association, but we're not saying the relationship is causal. There could be unexplained other mechanisms that we still have not unpacked or unraveled.
Okay. Thank you.
So there's
on that note, there's still work to be done, definitely. Thank you very much, Prof. This was a very good discussion. I'm sure it is. We've put some questions to the other researchers around Africa, and we'll probably be getting more people engaged in this question that you are pursuing. Thank you very much. I don't know if Nosipo, you have an announcement about when we have our next meeting, our next webinar.
Thank you everyone for joining, and thank you so much, Prof. Phumla. The next webinar is going to be on the 5th of June.
Okay.
On the
Thank you
5th
for
of June.
inviting me.
Thank you, Prof, for assessing the invites.
Thank you. Thank you, everyone. See you in June.
All right.
Thank you,
Have a
everyone.
good-
Bye.
Thank you. Thank you. Bye.